Hey guys, Ken is back again. Today I wanted to talk to you about the next group of large important biological molecules, and this is the nucleic acids. Okay, so DNA is probably the most famous version of the nucleic acids. And so if you look at the DNA, the Na part of this actually stands for nucleic acid. The D part is deoxy, which is the most famous type of nucleic acid, but there's another type of nucleic acid that's incredibly important for your body as well. And we'll talk a little bit more about that today as well. Okay. So as we go ahead and go through this, keep an idea that we're comparing this to the carbohydrates and lipids that were already talked about. And we'll go ahead and talk about proteins in our next lecture. So let's go ahead and move on and start looking at our nucleic acids. So our essential questions for today are, number one, what do DNA and RNA stand for? We already kind of talked a little bit about DNA, but let's clarify for our sake. And what are the differences between these two molecules? So if these are our two major types of nucleic acids, what separates them from each other? Number two, what are the structural pieces of the double helix? And we want you to be specific. This is an important structure that makes up DNA, and it relates to how DNA is going to be replicated. It relates to how DNA is going to be utilized to go ahead and make RNA for proteins. This is an important concept, and so we want to make sure that you understand the structure of DNA and why it's so amazing for all the things that DNA needs to do. And the third, what is Chargaff's rule and what does it tell us? Think about purines and pyrimidines, and we'll revisit these hopefully at the end. So let's start with just defining what DNA is. So DNA is deoxyribonucleic acid. And this is a nucleic acid that contains the genetic instructions used in development and functioning of all known living organisms. There are a few that may have RNA if you think about viruses, but are they alive or not? We kind of argued about that before. This chemical substance is present in the nucleus of all cells and in most living organisms. Now, we have to realize that there isn't a nucleus, and we'll talk more about this in a later class, in your prokaryotic cells. But the nucleus in general is the place where we go ahead and store our DNA. So what does the DNA do? Now, DNA has all the chemical changes that take place in the cells. It controls this by giving the instructions for it. It has the instructions to tell you what kind of cell is going to be formed. Is this going to be a muscle cell, a blood cell, a nerve cell? Even what type of organism it's going to be. Is this the cell of a buttercup or a giraffe or a salmon or even a human? All of these things are going to be determined by the DNA. But I really need you to think about DNA like the instructions that you're going to go ahead and get from IKEA. All right, they're really important if you're going to go ahead and assemble it, but they don't actually do the assembly. You do the assembly yourself. Now, just like when we were talking about carbs and lipids, we have both a polymer and a monomer structure. Remember, poly means many subunits, and mono means single subunit. And so in this particular case, the DNA molecule is one of the polymers, okay? And that DNA is made up of smaller subunits. Those smaller subunits are known as nucleotides, okay? And every nucleotide is actually made up of other pieces. There's a sugar in DNA, that is a deoxyribose, a phosphate group, PO4, and an organic base of some kind. There are 4 bases that we utilize in the DNA molecule: adenine, thymine, cytosine, guanine, and we'll talk a little bit more about that here in a minute. Okay, now when we switch over to RNA, which is the other major type of nucleic acid we're talking about today, there are some changes here, but we need to understand what that change is. Okay, when we talked about the structure of DNA on the previous slide, hopefully you realize that we have the deoxyribose, which is that sugar, we have the phosphate group, and we have the base, the nitrogenous base. Now, when we go ahead and look at RNA versus DNA, deoxyribose is the sugar in DNA, ribose is going to be the sugar in RNA. Okay, ribose is more like glucose. It's more of a smaller sugar. It's a monosaccharide, so is deoxyribose, but it has an extra oxygen on it. Okay, and we typically represent both of these molecules with what we call a pentose ring here. You can see this down at the bottom. When we go ahead and look at this in models, you'll see that there's 1, 2, 3, 4, 5 corners. Each one of these corners represents a carbon. Remember that for organic molecules, carbon is super important because it can make those 4 covalent bonds. Okay, this is still true when we start looking at nucleic acids. Now, both DNA and RNA utilize a model system that looks at the same shape. One of them representing ribose, one of them representing deoxyribose, depending on the type of nucleic acid. Now let's go ahead and move on and take a look at the bases. When we go ahead and start looking at the most common bases, in DNA, we're looking at adenine, thymine, cytosine, and guanine. Now, these are not the actual shapes of the molecules. I'll show you more of what that looks like here in a second. But when you start to look at these shapes that I've put here, what do you notice about them? Hopefully, you're recognizing that their shapes are what we call complementary and that they work together. If you go ahead and take a look at the shape for adenine, adenine is going to have this little triangle cut in here. And if you look at thymine, it has the opposite shape triangle section here. So the thymine is going to match up with the adenine. When we look at cytosine, you see this little curved section, curved in here. And when you look at guanine, you see this little curved out section here. So obviously cytosine binds with guanine. Now the bases are very specific in how they attach to each other. And this is not what actually causes them. They don't look like this, as I mentioned before, but shape is a really important reason why adenine fits to thymine and cytosine fits to guanine. Now, in each one of these pairs, we have one of two kinds of bases. We have a group of bases known as pyrimidines. These are going to be our single ring bases, thymine and cytosine. Each one has a ring of carbon and nitrogen atoms. You can see their structures here. This is compared to our two purines or double ring bases, which are adenine and guanine, and they have double rings you can see here. Now, when you go ahead and look at these structures, adenine binds with thymine, cytosine binds with guanine. Notice that there is one purine that bonds with one pyrimidine. Okay, so thymine is a pyrimidine. It binds with adenine, which is a purine. Cytosine is a pyrimidine that binds with guanine, which is a purine. Pyrimidine, the longer name, is for the smaller molecule. Purines, the smaller name, is for the larger molecule. Okay, so every pair is going to include one of each. So let's go over those nucleotides again. You have your purines, or double rings, adenine and guanine, and you have your pyrimidines, which are your single rings. These are cytosine and thymine. The structure that you're seeing here is going to be important when we start looking at how DNA fits together. You have down here on the bottom your phosphate groups. You can see them along here. And each phosphate is attached to our sugar. Now, if we're dealing with DNA nucleotides, see here it says DNA, this is going to be a deoxyribose sugar. So phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This makes up the backbone of the DNA molecule, and moving towards the center, we have these bases, okay? Your purines and your pyrimidines. All right, they're pointed inward, and then we have this chain out here of phosphate and sugar. All right, so let's go over the structure of a whole nucleotide again. In this particular case, we have a phosphate, a sugar, and a base. Let's start with the base. Can anybody go ahead and identify it? Okay, so if you said that it's the pentose ring you're seeing here, you are correct. This is your sugar. And in this case, we're telling you it's deoxyribose. So is this DNA or RNA? That's right. This is going to be for DNA. Now, we have two other parts to this molecule. What do you think is the phosphate? That's right. The PO4 is the phosphate group, so this is your phosphate. So we have our sugar and phosphate. These are the parts that are going to make up the backbone. Moving in here, we have thymine. Thymine is that's right, this is a base. So when we go ahead and look at this base, do we know if this is DNA or RNA? Well, because this is deoxyribose, we can be pretty sure that this is going to be DNA. But this ends up being an important thing because turns out thymine is only found in DNA. RNA doesn't have thymine but replaces it with a different nucleotide. All right. So once again, a molecule of DNA is a macromolecule or a polymer. So it has a whole bunch of these smaller subunits known as nucleotides. So several nucleotides joined together makes up a polymer. So if we go ahead and annotate here, we've got our phosphate group. Let me go ahead and turn off my pointer. We've got our phosphate group here. Oops, come on, wait a second. All right, still doesn't want to write for me. Come on. Technical difficulties. All right. So when we go ahead and do this, nope, it doesn't want to work. Try it this way. There we go. All right, so what you have here is one nucleotide, okay? You have your phosphate group, you have your sugar, and you have your base, okay? And when we chain one with another, with another, with another, this is going to go ahead and create that polymer. Again, we have phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, which we identified earlier as the sugar phosphate backbone. These pieces that are pointing towards the inside are the bases. Okay, so as we go ahead and move on, this is an important structure that we're going to go ahead and talk about. Now, let's move on and start looking at the actual structure of DNA. Now, DNA looks kind of like a twisted staircase. That's how a lot of people refer to it. And there are actually two strands that make up DNA. So you can see that over here on the left, you've got one strand here, and you've got your second strand here. And they twist upon each other. This is a structure that you may have heard of before. This is known as a double helix. Okay. Now this strand that runs along the outside is our sugar phosphate backbone or chain that we talked about before. So then these pieces that are in the center, do you remember what these are called? That's right. These are called the bases. All right. So let's go ahead and take a quick look at how this is going to go ahead and pan out. Now, here we go. We've got a long chain of nucleotides. So remember, nucleotides are the monomer, and a whole string of them is a polymer. But the DNA molecule is not a single strand, but a double strand. Okay, and when they bond together, again, notice that these complementary shapes fit together. All right, phosphate, sugar, phosphate, sugar, phosphate, sugar, so on and so forth. Couple things. Do you notice anything about those pentose sugars in this particular diagram? If you take a look, I'm going to go ahead and trace one on each side. Okay, now let's trace one on the other side. What do you notice? Okay, that's right. The one on this side is actually upside down compared to the one on this side. This is important to understand that the DNA molecule actually is going in opposite directions depending on which side you are on. And that these bases are always going to pair up in a predictable fashion where one section is going to go ahead and match up with its complementary. So we'll have adenine with thymine and cytosine with guanine. Okay? All right. So when we go ahead and start looking at this, Hopefully you guys recognize little dino DNA from Jurassic Park. Adenine is always going to pair with what? That's right, thymine. And cytosine is always going to pair with what? That's right, guanine. And very commonly, we will represent these with letters instead of with the actual full names. We have A for adenine, binds to T for thymine. C for cytosine, bonds to G for guanine. Okay? All right. Now, when we start looking at these pairs, there are reasons why they have to pair with each other. Now, in our little model here, we were just showing you that the shapes match. And shape is important, don't get me wrong. But if I were to go ahead and show you adenine with thymine and cytosine with guanine in these particular examples, do you notice anything else that's holding them together? Hopefully, you are recognizing that right here we have 1, 2 bonds, versus here we've got 1, 2, 3 bonds. This is really important because it's also another way that your body checks that adenine binds with thymine and cytosine binds with guanine. Okay. Now, does anybody know what kind of bonds hold this together? Okay, there are 3 types of bonds that you typically talk about in your physical science class. You have your covalent bond, you have your ionic bond, and one that you may or may not have talked about, which is a hydrogen bond. Okay, we'll talk more about this here in a second. All right, so how do we know that adenine and thymine must always bond with each other? Well, we've done a lot of experiments to go ahead and test this out. And this is what we typically would call Chargaff's rule. A gentleman by the name of Chargaff did studies of a whole bunch of different species, primarily of animals, and checked the amounts of these different bases. And what he discovered is that the percentage of adenine and thymine were almost always the same. And the percentage of cytosine and guanine were almost always the same. And this became Chargaff's rule. We now know, however, that this is because of the base pairing. Thymine binds to adenine because you have 1 purine and 1 pyrimidine. These are the shape issues that I was mentioning before. But in addition to that, you also have the actual bonding. There are only 2 bonds that are present between adenine and thymine versus the 3 bonds that are present between cytosine and guanine. So let's go back to that question of what kind of bond this is. If you go back and think about your bonds, when we look at a covalent bond, this is the strongest of all of the 3 bonds that we've mentioned here. There is a physical sharing of the electrons between the 2 parts of the molecules that connects the 2 objects together, making a very strong bond. The next strongest is what we call the ionic bond. In this case, one ion is going to lose an electron to the other ion, and then they're going to be attracted to each other by their opposite charges. So covalent's the strongest, then we move down to ionic. The weakest of these 3 bonds is actually the hydrogen bond, okay? Because it's actually attraction between partial charges because we're dealing with polar molecules. Do you remember we talked about polar molecules when we were going over water? Yeah, that's the uneven sharing of electrons. So why would we go ahead and use the weakest bond of the 3 to attach this very important molecule? Can anybody think of an answer? It's because there are millions of them. But at the same time, because they're weaker, they're easier to make and to break. And you have to break this thing apart every time you want to go ahead and make a copy. Anytime you're making a copy for protein synthesis, anytime you're making a copy to replicate the cell. So you have to be able to break this molecule apart and put it back together pretty easily. And for that reason, we go ahead and use hydrogen bonds so that we can do this, but because there are millions of them, it's still a very stable molecule. In this particular example, you're seeing cytosine and guanine. But because you have these 2 sugar phosphate backbones wound around the outside with the bases pointing towards the middle, we create this beautiful double helix structure. And this structure is probably what you're familiar with in terms of studying DNA. But if you zoom in on it, you can see exactly what it is we've been talking about. Okay? On the outside, you've got these two sugar phosphate backbones. You can see here I'm running right along the backbone. Okay? So if I start with the next one, I've got this backbone that goes right here and it twists around, and that's your sugars and phosphates. These little pieces pointing towards the center, these rungs of the ladder, if you will, are the bases. And notice they're not physically connected. They're attracted to each other by those hydrogen bonds that we just talked about. All right, so when we go ahead and start looking at this, this is your DNA structure again. This outside is the sugar phosphate backbone, phosphate, sugar, and because it's double-stranded like this, this is going to be for DNA, not RNA. And inside, we've got our bases. Okay, these are the bases, and you have cytosine and guanine, pyrimidine, purine, pyrimidine, and adenine's always with thymine. There's your 2 hydrogen bonds. Cytosine's always with guanine. There's your 3 hydrogen bonds. One sugar, phosphate, and base together is known as a nucleotide, and this is the monomer for our DNA polymer. Okay, so that's a lot. Now, before I let you go today, I wanted to go ahead and talk a little bit about the history of DNA. The structure of DNA was first identified by Watson and Crick back in 1953, and it's quite a story, actually. But it wouldn't have been possible if it wasn't for the work of 2 very unsung heroes, Maurice Wilkins and Rosalind Franklin. Now, Watson and Crick, because they came up with the structure, are the ones that got all the fame and fortune, but it was a picture taken by Rosalind Franklin that actually allowed Watson and Crick to go ahead and come up with this structure in the first place. All 4 of the characters are pretty unique in this story, and there's a video that we'll go ahead and watch that gives you a little bit more of the history and power of the discovery of the DNA double helix. But before I let you go, I want to talk a little bit about just how special DNA is. I know I talk about proteins as being the amazing thing because they do all the work, but the DNA molecule is a pretty cool marvel of structure. Okay? Now, if you stop to think about it for a second, each cell has about 2 meters of DNA if you line it up end to end. That's a ton when you stop to think about it. Okay, I am 6 feet tall, and that's about the length of me for one cell. Okay? And the average human has about 75 trillion cells by the time you're an adult. That's pretty impressive. So if you unravel all of the DNA and all the chromosomes in one of your cells, it stretches out to 2 meters. And if you do this to all the DNA in all of your cells, it would stretch from here to the sun and back about 400 times. That's pretty amazing. How do you fit that much material in that tiny amount of space? And the way you do that is by making the molecule incredibly thin. In fact, the diameter of DNA is 0.2, I don't even remember what this is. 1, 2, 3, 4, 5, 6, 7, 8, 9 decimal places meters. So 2 times 10 to the negative 9th meters long. That's pretty amazing. The distance between Earth and the Sun is about 93 million miles or 150 billion meters. So it's a long way. That is a ton of DNA. And for every single one of you, your DNA is unique. It's like a fingerprint that is used to identify just you. Okay? And even though it's not visible to the naked eye, it is unique to you. And we'll talk more about this, especially in our next semester. But the different arrangements of those nucleotides in a nucleic acid, especially one like DNA, provides the key to all the diversity that exists among all the organisms on our planet. We store that DNA in chromosomes, and you're probably used to seeing them as the little X's that they use in movies. That's only when they're condensed, but we'll talk more about that when we get to genetics. The code of life, or this code in the chromosomes, is specified based on the order of the bases. And that's written in those 4 nucleotides bases that we talked about, nucleic acids, right? Have these bases: adenine, thymine, cytosine, and guanine, and depending on the order and sequence of those bases, you determine what proteins are produced and what that organism looks like. So I'm going to leave you today with one last little table. And when we go ahead and start looking at how much variation exists in different species, how many chromosomes they have, how much DNA they contain, one of the misnomers that a lot of people tell me is that, oh, well, we have more chromosomes, that's why we're more complex. Well, if that were the case, I encourage you to go ahead and take a look at goldfish. Okay? The goldfish here have 94 chromosomes. Does that mean they are the most advanced species on this list? I don't know that that's necessarily the case. When we go ahead and look at this list, however, it does tell us something pretty spectacular. Okay? When we go ahead and look at these, what do you notice about all these numbers? 46, 48, 48, 60, 38, 94, 8. What do they all have in common? If you said that they're all even, you would be right. Okay? These are all sexually reproducing organisms, and so they get genetic material from both sets of parents, okay, from their mother and their father, and because they get a set, it has to be going ahead and being divisible by 2. Now, some of you at this point, as I'm talking, may have noticed down here when we start looking at the orange. Whoa, that's got 27. That's not even. But if you go ahead and take the other 2 genetic numbers that we have here, 36 and 18, if you divide them both in half, what do you get? Well, half of 18 is what? That's right, 9. And half of 36 is what? That's right, 18. So if you take 18 plus 9, what do you get? That's right, 27. So this is actually a hybrid species. So it's interesting when we start looking at these chromosome numbers. Now think about what we talked about with the mule as well, being a cross between a donkey and a horse. And maybe you can kind of make heads or tails out of that right now. So chromosomes are where we store the DNA. DNA is one of 2 nucleic acids that we have that we're going to discuss in this class. DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. DNA and RNA are both made of smaller subunits or monomers known as nucleotides. And when we go ahead and start looking at this, DNA is 2 strands wound up in what's called a double helix versus RNA, which is going to be single-stranded. Now, your worksheet's going to go over these similarities and differences in a little bit more detail, but I hope this is a good introduction, you guys, to your nucleic acids. Thanks for sticking with me. Hope you have a good one. Take care of yourselves. Bye-bye.